Welcome to the PubMed Daily Review for April 12th, 2026. Today we scanned 2,792 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Neurological Disorders and Perception. This trend explores how neurological conditions impact perception and how interventions can be developed and assessed. This cluster covers 711 papers. Representative work includes evolution of drug development trends in multiple sclerosis, analysis of mainland China and global landscapes from 2004 to 2024, principles of gamma synchrony predict figure-ground perception in texture stimuli, and stimulus dependencies rather than next-word prediction can explain pre-onset brain encoding in naturalistic listening designs. Trend 2 of 5, Microbial Pathogens and Evolution. This trend explores microbial evolution, pathogenesis, and their impact on host health and disease. This cluster covers 615 papers. Representative work includes reducing mapping reference and lineage bias in Mycobacterium tuberculosis, comparable efficacy of bictegravir/emtricitabine/tenofovir alafenamide and doravirine/lamivudine/tenofovir disoproxyl fumarate in therapy-naive human immunodeficiency virus adults with very high baseline viremia, and a comparison of routine blood culture methods and multiplex quantitative PCR for detecting pathogens in simulated polymicrobial blood cultures. Trend 3 of 5, Materials Science and Ecology. This trend explores how materials science innovations interact with and impact ecological systems and processes. This cluster covers 459 papers. Representative work includes comparison of magnetic nanoparticle immobilization methods using MPS and MRX. Carotenoid aggregates negatively impact chlorophyll levels and disrupt chloroplast development in peaches, and active polymers containing silver nanoparticles combined with formulations based on essential oils. Quality effect on cereals and dairy products. Trend 4 of 5, Clinical Research and Health. This trend explores diverse clinical research applications, from therapeutic outcomes and diagnostic methods to patient perspectives and anatomical insights. This cluster covers 608 papers. Representative work includes omalizumab therapy results in defined behavioral changes and improvements in quality of life in solar urticaria, mapping the global surge in postoperative sleep research from 2014 to 2024, bibliometric analysis, and in memoriam. Professor Jose Cunha-Vaz, 1938 to 2026. Trend 5 of 5, Cancer Biology and Treatment. These papers investigate molecular mechanisms, genetic factors, and therapeutic strategies impacting various cancer types. This cluster covers 399 papers. Representative work includes loss of ZNRF3/RNF43 unleashes EGFR in cancer, comparing the prognosis of non-small cell lung cancer patients who did not undergo surgery after neoadjuvant chemotherapy combined with immunotherapy. A retrospective study, and retraction. Influences of S100A8 and S100A9 on proliferation of nasopharyngeal carcinoma cells through PI3K/ACT signaling pathway. Up next, our first paper out of 5, Discovery of SKP2, Recruiting PROTAC for Target Protein Degradation by Guanjuan Dong and Jianwei Chun from School of Pharmaceutical Sciences, Shenzhen campus of Sun Yat-sen University, Shenzhen, PR, China. Let's break down its key contributions. Targeted protein degradation, or TPD, is a revolutionary therapeutic approach. It uses specialized molecules to tag protein for destruction by the cell's own machinery. While promising, we've been limited by the small number of E3 ligases we can effectively recruit. This research investigates whether SKP2, a protein often found in high levels in cancer cells, can be a new player in this degradation game. To explore SKP2's potential, the researchers designed and synthesized novel PROTACs. They used computational methods like molecular docking to guide their design, and then validated the binding affinity using surface plasmon resonance. Key experiments involved Western blotting to confirm protein degradation and co-immunoprecipitation to understand the interactions. The study focused on using these PROTACs in cancer cell lines to degrade target proteins like BRD4 and the androgen receptor. The results were highly encouraging. The most potent PROTAC, compound 2:1, achieved an impressive 99.6% degradation of BRD4 at a 10 micromolar concentration. The binding affinity of the SKP2 recruiter SL1 to SKP2 was quantified at 1.81 micromolar. Furthermore, these SKP2-based PROTACs effectively degraded the androgen receptor in relevant cell lines, validating the approach. This study highlights a significant advancement. SKP2 can indeed be effectively utilized for targeted protein degradation via non-covalent PROTACs. This discovery broadens the range of E3 ligases we can employ, offering new avenues for drug development, especially for targets previously considered difficult to degrade. While promising, further studies are essential to refine these PROTACs and evaluate their potential in clinical settings. Up next, our second paper out of 5, discovery of a first-in-class Murine -2-recruiting positive transcription elongation factor B PROTAC degrader with selective anti-tumor activity by Xian Guan and Xian Han from Cancer Institute, Key Laboratory of Cancer Prevention and Intervention, China National Ministry of Education, of the Second Affiliated Hospital and Institute of Translational Medicine, Zhejiang University School of Medicine, Hangzhou, China. Let's break down its key contributions. The positive transcription elongation factor B, or PTFB, complex, which includes CDK9, plays a vital role in gene transcription and is a key driver of cancer cell growth. While targeting CDK9 is attractive for cancer therapy, developing selective and safe inhibitors has been difficult. This study explores a new strategy using PROTAC technology to achieve targeted degradation of CDK9. Researchers designed novel PROTAC molecules by linking a known CDK9 inhibitor to a ligand that recruits the MDM2-E3 ligase. They synthesized and tested a series of these compounds in various cancer cell lines. The lead compound, DSDK9-010, was found to effectively degrade CDK9 and its partners, leading to downstream effects on transcription and the p53 pathway. The lead compound, DSDK9-010, showed remarkable efficacy. It potently degraded CDK9 and its partners in cancer cells, with IC50 values in the submicromolar range for inhibiting cell growth. Importantly, in animal studies, DSDK9-010 significantly suppressed tumor growth in lung cancer and Ewing sarcoma models, all while maintaining an excellent safety profile. This study establishes DSDK9-010 as a novel, first-in-class PROTAC degrader that effectively targets CDK9. Its unique mechanism, which disrupts transcription and activates the p53 pathway, leads to selective killing of cancer cells. This approach holds significant promise as a new therapeutic strategy for cancers with wild-type p53. Up next, our third paper out of 5. The non-vesicular cholesterol transporter GRAMD1C is a pan-coronavirus antiviral target by Jolene Su and Melissa Vazquez Hernandez from State Key Laboratory of Agricultural Microbiology, College of Veterinary Medicine, Huazhong Agricultural University, Wuhan, Hubei PR, China. Let's break down its key contributions. Coronaviruses like SARS-CoV-2 cause significant global health issues. These viruses are masters at manipulating our own cellular machinery, particularly lipid metabolism, to help them multiply. While we know cholesterol plays a role, the precise host factors involved in building the virus's replication factories are still being uncovered. This research dives into one such factor, GRAMD1C, a protein involved in transporting cholesterol. To understand GRAMD1C's role, the researchers used a variety of techniques. They created cell lines where GRAMD1C was either removed or overproduced using CRISPR technology. They then infected these cells with different coronaviruses and measured viral replication using methods like Western blots and quantitative PCR. They also used advanced microscopy to visualize viral structures and protein interaction assays to see how GRAMD1C interacts with viral components. Finally, they tested the effect in animal models. The results were striking. Removing GRAMD1C significantly hampered the replication of the TGV virus. More importantly, this effect wasn't limited to one virus. GRAMD1C knockout cells showed reduced replication for viruses from alpha, beta, and delta genera, including SARS-CoV-2. Mechanistically, the study found that GRAMD1C interacts with key viral proteins and is essential for forming the double membrane vesicles where viruses replicate. This role was also confirmed in mouse models, where GRAMD1C deficiency led to lower viral loads. This study identifies GRAMD1C as a critical host factor and a promising, broad-spectrum target for developing new antiviral drugs against coronaviruses. By interfering with cholesterol transport and the formation of viral replication sites, targeting Gram1C could offer a new strategy to combat a wide range of COV infections. While these findings are exciting, further research is essential to develop safe and effective inhibitors for clinical use. Up next, our fourth paper out of 5. NR2F6 deletion revives CAR T-cell function and induces antigen-agnostic immune memory in solid tumors by Dominik Humer and Gottfried Bayer from Cell Genetics, Medical University of Innsbruck, Austria. Let's break down its key contributions. CAR T-cell therapy has revolutionized cancer treatment, showing remarkable success in blood cancers. However, its application in solid tumors faces significant hurdles. The tumor microenvironment and the diverse nature of tumor antigens often lead to CAR T-cell exhaustion, limiting their effectiveness. This research explores a novel genetic modification to overcome these limitations. To investigate the role of NR2F6, researchers used advanced gene editing techniques in mouse models of solid tumors. They employed CRISPR/Cas9 to delete the NR2F6 gene in CAR T cells, then administered these modified cells to mice bearing tumors. Various in vitro and in vivo assays were used to assess CAR T-cell function, persistence, and overall anti-tumor response, focusing on the mechanisms behind any observed improvements. The results were striking. Mice treated with NR2F6 deleted CAR T-cells showed significantly reduced tumor growth and a remarkable increase in complete remission rates compared to controls. These modified CAR T-cells maintained a progenitor-like state, produced higher levels of key effector cytokines like IFN and granzyme B, and crucially, induced a durable, antigen-agnostic immune memory that persisted even after the engineered CAR T-cells were no longer detectable. This study highlights NR2F6 as a critical intracellular checkpoint that, when inhibited, can reprogram CAR T-cells for superior performance in solid tumors. The induction of durable, antigen-agnostic immune memory is particularly exciting, offering a potential solution to immune escape mechanisms. These findings pave the way for novel CAR T engineering strategies that could significantly improve outcomes for patients with solid malignancies. Up next, our fifth paper out of 5. A photodynamically activated nanoplatform relieves glucose-driven immunosuppression to potentiate STING immunotherapy in triple-negative breast cancer by Yongxin Long and Weijie Zhang from A Division of Breast Surgery, Department of General Surgery, Nanjing Drum Tower Hospital, Affiliated Hospital of Medical School, Nanjing University, Nanjing, Jiangsu, 210000, China. Let's break down its key contributions. Triple-negative breast cancer, or TNBC, is a particularly aggressive form of breast cancer with limited treatment options and a high risk of recurrence. A major challenge in treating TNBC is its ability to create a shield around itself, an immunosuppressive tumor microenvironment. This environment is fueled, in part, by how cancer cells process glucose. We aim to develop a novel nanoplatform to break down this shield and boost the body's own immune response against TNBC. To tackle this challenge, we designed a sophisticated nanoparticle called DI/SU6 at STF. This nanoparticle acts as a multi-pronged attack system. It carries a STING agonist to activate the immune system, a photosensitizer for photodynamic therapy, and a GLUT1 inhibitor to disrupt the cancer cell's glucose supply. We tested this system in both cell cultures and a mouse model of TNBC. Our results were very promising. When activated by a laser, the DI/SU6 at STF nanoplatform significantly increased cancer cell death through apoptosis, showing a 1.82-fold increase compared to a control group. We also observed a substantial generation of reactive oxygen species, a key component of photodynamic therapy. Importantly, the nanoparticles showed excellent accumulation in tumor tissues, reaching 2.1 times higher fluorescence than controls, while minimizing uptake in healthy organs. This all translated to potent inhibition of tumor growth. In conclusion, this study demonstrates a powerful new strategy for treating triple-negative breast cancer. By cleverly combining photodynamic therapy with metabolic intervention and STING activation, our DI/SU6 at STF nanoplatform effectively reprogrammed the immunosuppressive tumor microenvironment. This approach holds significant promise for overcoming treatment resistance and offers a potential new avenue for patients with TNBC. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.